Okay, good morning and good afternoon, ladies and gentlemen. Thank you for joining us today in our webinar series that is entitled Industrial Webinar QC in Microbiology. So today we are in week two, and we are actually very pleased to announce that we have Professor Yuk himself who will be joining us live today to answer some of the questions from last week as well as this week's lecture. So again, just as a reminder that this webinar series will consist of four separate lectures, all of them delivered by Dr. Yuk. So this week, we actually have a lecture that is entitled Rapid Detection for Total Bacteria Count. And please do join us next week again on the same day, Wednesday, at the same time, and we will have a lecture on microorganisms that are related to juice products. Again, today, just as a reminder, because Professor Yuk will be joining us live, you can actually type in your questions and we will address them at the end of the session. And again, today we will actually be conducting some surveys, and you can actually get an instant certification for your attendance in the program by completing the survey. And to do that, you can either scan the QR code that will be flashed later, or you can also check the announcement icon on your browser, on your GoToWebinar platform to actually get the direct URL link. So I will again flash this towards the end of the presentation and the webinar so that you can actually complete the survey and get your certificate. And lastly, this is our instant certificate of participation that again can be obtained by completing our survey. So without further ado, again, I would like to introduce our esteemed speaker. So Professor Yu is actually currently a professor at the Department of Food Science and Technology, Korea National University of Transportation. Prior to taking up this position in South Korea, he was actually an assistant professor at the Department of Food Science and Technology at the National University of Singapore. Dr. Yu is a very avid researcher and a respected expert in the field of food science, having published more than 100 peer-reviewed publications. He is also currently an active member of the International Association of Food Protection. So today he will be delivering a lecture on rapid detection of bacteria count.
Hi, I'm Hyun Yuk from Korean National University of Transportation. In this webinar, I'm going to introduce some rapid detection method for total bacterial count. As you may know, total bacterial count is the most widely used quality indicator at food manufacturers. This is because the number of total bacterial cells in food products may indicate the degree of food spoilage and microbial contamination. So it is very important to count bacterial cells in food products to see if this number meets the microbiological criteria. The most common way to count bacterial numbers is based on traditional culture method. But this method is time-consuming and labor-intensive, so it is difficult to take immediate action to correct the microbiological problem in the final products under HACCP plan. For this reason, some scientist companies have tried to develop rapid method to count bacterial cells in food products. So I will try to cover newly developed rapid method for total bacterial count in this webinar. As you can see this figure, the rapid method for total bacterial counts can be divided into two types, which are indirect culture-based method and non-culture-based method. The indirect culture-based method still requires bacterial cell culture to count the number of cells in food products. On the other hand, non-culture-based method does not require cell culture, so this rapid method will be better than the indirect culture-based method in terms of the time required for microbiological analysis. Different types of detection techniques are included in each category of rapid method. Results cell viability, miniaturized most probable number method, and bacterial metabolite analysis such as carbon dioxide and oxygen are included in the indirect culture-based method. For the non-culture-based method, 5 technologies have been developed so far, and they are ATP assay, direct EPI fluorescent filter technique, flow cytometry, impedance technology, and PCR-based method. In this category, most technologies require specific equipment to detect bacterial metabolites or bacterial cells themselves. Now I'm going to introduce each method more detail next slide. The first direct method is Resazurin cell viability assay. Resazurin is a chemical compound which is a cell permeable redox indicator. Resazurin is very effective chemical to monitor viral cell numbers. Resazurin itself is non-fluorescent blue dye, but this can be converted into pink fluorescent resorupin in the presence of NADH, which is a reducing agent in metabolically active cells. NADH is metabolite which is produced during energy-yielding metabolism, such as 3-carboxyl acid cycle, which is called the TCA cycle. This figure shows the principle of Resazurin cell viability assay. Once we add blue Resazurin solution into the sample containing bacterial cells, NADH dehydrogenase, which is produced from live cells, transfers electrons from NADH to Resazurin, resulting in the reduction of Resazurin to resorupin, which is a pink color of compound. In this reaction, NADH acts as a cofactor of NADH dehydrogenase. NADH and NADH dehydrogenase are found in metabolically active cells, as I mentioned before, so this reaction can be found only in live cells. Let me show an example of this detection process. As you can see this figure, we add Resazurin solution into 96 wells containing bacterial cells and incubate them for a while. If they are live cells, the color of solution is changed from blue to pink, so we can quantify the intensity of color to convert it to the cell numbers if we use microplate leader at specific wavelengths. It can be done within 4 hours, which is more quickly done compared with the total plate count method. The second method is using most probable number called MPN. MPN method is already established method which are widely used to quantify coliforms and E. coli. Miniaturized MPN is just the small size of traditional MPN method, so detection using MPN can be carried out in a specific instrument. So you need to understand the principle of MPN method first. Cell count using MPN is estimating bacterial cell number based on statistically probabilities like Gross probability. Traditional MPN method uses multiple glass test tubes and count the number of test tubes which show positive for bacterial cell growth. However, this method is not practical for multiple assays. So if this MPN method could be miniaturized and the cell growth could be detected by microplate leader, we can save time and labors. In this miniaturized MPN method, target bacteria multiply in the culture medium and produce signals based on fluorescent pH indicator and beta-galactosidase activity, which are easily detected by microplate reader at specific wavelengths. If the metabolite for specific target microorganism is already known, we can apply this method to the different types of microorganisms, such as total bacterial count, coliforms, and yeast and molds. Here is one example of miniaturized ambient test kit. As you can see this photo, there are some small size of tubes in the kit. We add the sample into each tube and incubate them for 18 to 24 hours. After incubation, microplate reader can read the gross bacterial cells in each tube based on turbidity or a specific fluorescent indicator. Right side of figures shows the correlation between traditional ambient test, which is an official method, and commercial miniaturized ambient test. As you can see this data, the accuracy of commercial miniaturized ambient test is very close to the official ambient test. So it can be very useful when you want to determine the microbial contamination using ambient test. The third one is the method to count bacterial number by detecting carbon dioxide. Carbon dioxide is a universal metabolite produced by most microorganisms. So there is a correlation between the concentration of carbon dioxide and bacterial count. This concept is a key point for this detection technology. As you can see the left figure, the vial has its own sensor on its bottom side. Once microbial cell produce carbon dioxide by fermentation, these carbon dioxides will be detected by that sensor. So concentration of carbon dioxide could be quantified by the sensor, indicating the degree of microbial contamination. The right figure shows the difference in the bottom of vials between no growth and growth of microorganisms. So we can easily recognize the difference between them using fluorescent sensor. However, there is one limitation for this detection technology. If non-fermenting microorganisms are contaminated in food samples, these microorganisms could not be detected by this vial because there will be no carbon dioxide generated from these microorganisms. Next one is the detection technology, which can detect oxygen level in the sample. The principle of this detection technology is based on the fact that microorganisms consume oxygen when they produce adenosine triphosphate, which is called ATP, during energy metabolism. All aerobic and facultative anaerobic microorganisms need oxygen as the final electron acceptor in the electron transport system to produce ATP, like a human. This fluorescence-based technology is using oxygen sensing probe, which can detect bacterial cells by monitoring bacterial respiration. So this system measures bacterial oxygen consumption for respiration and equates that to viable microbial numbers. The right side of figure is one example of detection technology based on oxygen consumption. According to this company, total viable cell count level in raw meats of about 6 log CFU per gram can be detected within 5 hours, indicating sample preparation time. Including sample preparation time. However, this technology also can be useful only for aerobic or facultative anaerobic microorganisms, as I mentioned before, because anaerobic microorganisms do not need oxygen to produce energy. Okay, these 4 indirect culture-based methods are currently available at the market. Now I'm going to introduce some non-culture-based detection methods for microbial contamination. The first method is ATP detection method. This technology has widely used at food manufacturers to check the degree of microbial contamination on equipment or food contact surface. As I mentioned before, ATP is energy molecules which are produced by the cell respiration, and these molecules disappears within 2 hours after cell death. So ATP is kind of indicator of cell viability. High level of ATP concentration indicate poor sanitation practices and the potential for post-process contamination. But please note that all live cells, including human and animals, also have ATP. So sometimes non-microbial ATP like ATP from the meat product can be detected, which makes some errors in this assay. For this reason, this method is used for monitoring purpose, but not used to indicate numbers of microorganisms. The detection of ATP itself is very rapid and simple, so you can detect ATP from swab samples within 1 to 3 minutes. So you don't need to culture the sample for ATP detection. Therefore, immediate action can be taken if you detect high concentration of ATP from food contact surface after sanitation. Now let me explain how it works. As you can see this figure, ATP is a cofactor of luciferase, which is an enzyme from firefly. When luciferase oxidizes luciferin, which is substrate, ATP participates in this reaction to help the enzyme for oxidation reduction reaction. This enzyme reaction produces light, which can be detected by the device, which is called a luminometer. So the quantity of this light is proportional to the quantity of luciferin oxidized. The right figure shows the relationship between cell counts and the concentration of ATP, which indicates the higher number of microorganisms, the higher the ATP concentration. The second detection method is using direct Fe fluorescent filter technique, which is called DEFT. This method is useful for liquid sample. A liquid sample like a juice and milk is filtered by polycarbonate filter, and then the filter is stained with a fluorescent dye like acridine orange, followed by examination using Fe fluorescence microscopy, as you can see the right photo. Acridine orange binds to DNA, which is shown as green color, or RNA is shown as orange color. RNA is kind of indicator for cell viability, so orange colored cells are live and green colored cells are dead, as you can see the left side of figure. So we can differentiate live cells from dead cells by these fluorescent colors. As I mentioned before, this technique is only for liquid sample but not for solid sample. And another limitation is that more than 6 log cells per milliliter can be detected, so detection limit is a bit high. The third detection method is using flow cytometry. Flow cytometry is kind of a cell counter at single cell level, so we can use this equipment to count bacterial cells, which is stained by a fluorescent dye, as you can see the right side figure. This method is also useful for liquid samples such as juice, soft drink, and milk like DEFT. Because this instrument can detect cells in the sample directly, no enrichment of sample is required, and we know the degree of microbial contamination within a few hours. This method has been validated in some previous studies. I give you an example. This study has tried to validate the flow cytometry technique to count E. coli. For this, the investigators compared the number of E. coli cells by plate count method with those by flow cytometry. As you can see in the left figure, it is not doubt there is a correlation in the count of E. coli between plate count method and flow cytometry. Another try has been made, as you can see in the right figure. These investigators have tried to validate the flow cytometry based on enumeration of live bacteria cells, which are stained with live dead bacterial viability kits. In this study, this investigator collected 31 samples and analyzed them by both flow cytometry and plate count method. According to their results, all test samples fits reasonably well with the overall standard curve obtained for live E. coli, except for one outlier. Therefore, this data suggests that the results obtained from flow cytometry are very reliable in terms of count of live bacterial cells. The first rapid detection method is using impedance technology. This technology can count bacterial cells based on the measurement of changes in electrical resistance when cells pass through the micropore, as you can see the right figure as one of examples. Because resistance occurred from passing the cell through the pore is measured, the impedance technology does not require any fluorescent dye like other rapid detection technologies. That is why we call this technology as label-free technology. Because current impedance technology can detect very small amount of electrical change, this technology has been widely used for development of biosensor. I give an example for how the bacterial count based on impedance technology correlates with plating count. This table shows the correlation between number of lactobacillus strains or species and correlation coefficient between plating count and impedance result. As you can see this table, there is a correlation in the bacterial count results between impedance technology and plating count, which shows more than minus 0.97 of correlation coefficient when single strain cultures were used. However, the coefficients decreased when mixed strains belonging to the same species or belonging to the different species. So this data suggests that it is necessary to carry out the calibration of the impedance system separately for each category of food to determine the total viral cell counts, because each food consists of different microbiota. Therefore, it is difficult to apply this impedance technology for various foods for total viral cell counts at this moment. I think this technology will be consistently improved through the researches. The last technology that I want to introduce is PCR-based detection technique. PCR stands for polymerase chain reaction, which can detect specific genes in bacterial cells by amplifying the genes using DNA polymerase. PCR instruments have been well developed, and we can find many of them at the market. For total viral cell count in foods, PCR-based quantitative assay targets highly conserved gene sequences like a 16S ribosomal RNA, TUF gene, RPOB gene, or etc., which are common to almost all species of bacteria. Because gene copy numbers are closely related to number of bacterial cells, PCR-based method can generate quantitative results, and it can be done within about 2 hours if we use real-time PCR machine. So this PCR-based method is more rapid than plating method. There are two limitations for using PCR-based method for total viral cell counts. Firstly, this method is not suitable for precise quantification of bacterial counts because the copy number of each specific gene varies by among bacterial strains. Secondly, this method cannot differentiate live cells from dead cells because bacterial DNA can still exist in dead cells. To overcome this drawback, fluorescent photoaffinity label method has been developed, which use either EMA or PMA as fluorescent particles. The principle is very simple. If cells are viable, cell has intact membrane, which prevent the entry of EMA or PMA. So DNA can be amplified by PCR. However, if cells are dead, EMA or PMA can enter inside the cell through the broken membrane and bind the DNA, which prevent DNA amplification. Based on this principle, live cells can only be detected by PCR. Sorry. Let me show you an example. This figure shows the standard curve for quantification of bacteria in ready-to-eat foods, fruits, and vegetables detected by PCR. X-axis indicates the bacterial count by plating method, and the Y-axis indicates CT value. CT value means the cycle threshold, which is defined as the number of cycles required for the fluorescent signal to cross the threshold. So lower CT values means that there are more bacterial cells in the sample. In this figure, the line indicates the count result from plating method, and the dots indicate the CT values obtained from PCR. R square value was about 0.9, so it seems that the correlation in the cell number result between plating method and PCR is a bit strong. However, it still needs to be improved for the increase of correlation between plating method and PCR. Another point in this data is the detection limit. As you can see, x-axis, the detection limit is about 4 log, which is much higher than plating method. In general, detection limit of PCR is about 3 logs, so lower than 2 logs cannot be detected by PCR. That is why this PCR method is not suitable for precise quantification of bacterial count in food samples. Okay, so far we have a look at the rapid detection method based on the indirect culture and non-culture to enumerate bacterial cells in foods. Also, I have covered their principles as well as their pros and cons. Now I'm going to summarize this presentation. The reason why these rapid detection technologies have been developed is that traditional culture-based methods for total bacterial counts are time-consuming and labor-intensive. For the immediate action to be taken, the application of rapid detection method is crucial. There are 3 types of indirect culture-based method, which are Rezazurin cell viability, miniaturized MPN, and assay of bacterial metabolites like carbon dioxide and oxygen. These methods are relatively rapid compared with traditional culture-based method, but they still require culture of food sample. Non-culture-based detection methods include ATP assay, DEFT, flow cytometry, impedance technology, and PCR-based method is more rapid than traditional and indirect culture-based method. However, these methods are more expensive than traditional and indirect culture-based method because they need special instruments. Another problem is that these methods are not adopted by the regulatory agencies, so this method can be used only internally. However, I expect that these rapid detection methods will be used routinely by food companies in near future because these technologies are continuing to be improved and food companies want to know the result of microbial tests as quickly as possible. Okay, this is what I have for this webinar. If you have any question, please contact Hazel Lim. She will forward your questions to me. Thank you very much for your attention.
Okay, thank you so much for joining us live today. So we do have quite a number of questions actually. Yeah, very interesting actually indeed. So we will just start off with the two questions that we had from last week from the week one lecture. So the first question is, how do we establish a protocol for an accelerated shelf life study? For example, a long shelf life cake with 6 months expiration placed in a 50 degrees Celsius incubator will be equivalent to how many weeks or months at room temperature?
Okay, thank you very much for the asking this question. Um, well, I think this question is related to my first webinar, but to be honest, I don't have any experience about accelerated shelf life study, and I have no idea how to conduct this study. But for this question, I have searched some literatures and protocols, but there is no direct answers for this question. With my understanding on the principles of accelerated shelf life study, it is difficult to answer on this question because individual food should be tested with its own accelerated shelf life study with some mathematical modeling to see how long it is stable in terms of microbiological, physical, and chemical properties. So to get better answer for this question, I suggest you the person who wanted to get this answer may obtain the protocols for the specific food, like a cake, from the literatures and protocols.
Sure.
Yeah,
Okay.
that's my
Thank you so much. Sure.
sorry.
Okay. Thank you. No problems. Thank you for the answer. Okay. So we'll just move on to the second question because of time limitation. So if a spoiled food with an unknown source of contamination is to be tested for pathogen identification, how do we decide which of the four methods that you talked about in your first lecture is to be employed, and how and or do you always use all of them to get a general idea of the pathogen identity?
Okay, I think this is very good question, and this question is just directly my first webinar.
Hmm.
Um, as I mentioned in my previous webinar, there are three microbiological indicators like quality, hygiene, and safety indicators. Actually, quality indicator like a total bacterial counts is not directly correlated with the presence of specific pathogens. But hygiene indicators such as E. coli and the food sample E. coli are indirectly correlated with the presence of specific pathogens like E. coli, like pathogenic E. coli and the Salmonella, because they are generally found in the lower intestinal tract of animals. So if you find that positive result of E. coli in your food samples,
Hmm.
you may test the food sample using a specific pathogen protocol to identify which pathogen is present in that food sample. But you should aware that some foodborne pathogens such as Staphylococcus aureus and Bacillus cereus are not correlated with the fecal contamination. Another way, you may refer to the result of safety indicators such as Listeria species detection and thermostable deoxyribonuclease, which we call that TNase. If you get the positive result of Listeria species in your food sample, it is likely that Listeria monocytogenes is present in the sample.
Hmm.
Also, if your TNase is found in the sample, it is likely that enterotoxin, Staphylococcus aureus, is present in that sample. This is just my answer for the question, but if you really don't know which pathogen you should detect for the your food sample, I suggest you can check the microbiological standard for your country or for exporting country. I think that is the correct way. Thank you.
Mm. Okay, thank you, Dr. Yu, for the answer. Okay, so we'll move on to the questions that we do have for today. The first one is for the rhesus urine test. How do you ensure that the sample actually remains pink and not become non-fluorescent, which is a result of the formation of dehydroresorption?
Okay, this is very good question. Thank you very much. As you can see, my this webinar, the presentation slide, there's two steps. Okay, first one is very blue color of resazurine is converted to the resolution by the NADH dehydrogenase, and this the material chemicals become pink color. And
Mm,
also the we leave the further, the reactions. They also resolution is dehydrogenase, dehydrolyzed to the dehydroresorpine, so which is a colorless material. So I think that's key point is the time. If your just reaction is taking about 3-4 hours, we can see easily the pink color if they are live cells. But if you leave more than that, this pink color disappear. So you can set
mm,
up the how long your reaction is occurs.
mm. I see. Okay. Thank you, Dr. Will. Okay. Hopefully that answers the question. Okay. And then the next question is, besides ATP measurement, which other rapid detection method is commonly adopted in the food industry?
Okay, I think the person who asked you this question, he or she want to know the which detection, like ATP measurement, to use
Mm,
in the spot in the field. But
yeah.
as far as I know, the ATP assay is good assay. I don't
Mm,
know other methods like ATP measurement, because this ATP detection is very rapid, which is I can detect the ATP the contents in the within the 1 to 3 minutes. But there's no other method to detect the microcontaminants like this.
mm, I see. Okay. Thank you, Dr. Yu. Okay. The next question is, could you please elaborate further about the limitations of flow cytometry in the detection of bacterial count?
Okay, thank you.
Mm,
Flow cytometry. Actually, we can use that in flow cytometry not only for the detect microbial cell, also we can use that the some not mathematics, sorry, mechanism
mm,
study. Okay, so the I already mentioned in my webinar for the principle of flow cytometry. This detect the fluorescent light for the individual cells. So the like an animal cell is very big. We can easily spread them in the sample solutions, but bacterial cells sometimes they're aggregated together.
mm,
So in that case, we can detect a right number of bacterial cell by flow cytometry. That's I think a limitation of this technology. And so I suggested the it's very be careful that when you prepare the samples. So you
mm,
can just using the protection to spread the cell very well.
mm.
The
Right.
second limitation, that's just some legal problem. Okay, so far there's no any other regulatory agency to adopt this flow cytometer result.
Mm,
So still they require the actual number of bacterial cells on the agar medium. So, but
mm.
I don't know. In the future, maybe they will be solved.
Right. Okay. Okay. And with regards to the regulatory issues that you're talking about, there are actually two related questions. Maybe we'll just try and address those ones first. So why do you think that regulatory agencies are not really willing to accept the data? I think you did mention that they actually wanted the actual number at the period. But apart from that, is there any other reasons, at least
Any
in
other?
Korea?
Sorry.
at least in Korea, in your country, is there any reason, any other reason why regulatory
Oh,
agencies are not keen to accept
yeah, yeah,
data from
yeah.
FCM?
That's a good question. Thank you.
Yeah.
Um,
Mm,
okay, so it's same as us. It's the same all the country. So as you can
mm,
see the food regulation in Singapore.
yeah.
There's a standard, a microbiological standard for each specific food. So
Mm,
in that case, well, it's the same as Korea. It shows that, like, for example, orange juice, not that the total bacterial count is not more than 50,000 CFU, something like that.
mm,
So in the micro standard itself, they show that number of bacterial cells. But actually, the flow cytometry is that we can just we try to quantify the number, but that this number not exactly same as actual number, because
mm,
this flow cytometry check the individual cells, but its the colony is totally different concept with the individual cell. The colony is more than they aggregate more than one cells.
mm,
So the regulation viewpoint, the microbial standard show the number of CFU colony forming unit, but flow cytometry just show that number of cells.
oh, okay.
So this is something kind of mismatch
Yeah,
between the
I
results.
see. I see. And there's not much data in the literature to support the actual individual cell
Right,
count, I
right.
see.
Yes, yes.
I see. Right,
So
right. That
if you
makes
see
sense.
that one colony on the plate count,
Mm,
one colony on the plate. So
yeah.
when you put in the flow cytometer, it's we can see that more than one cells.
Yes. Okay. Understood. Okay. Thank you for that, Dr. Yu. And then the next one that is also regulatory related is which of the rapid detection method is actually more promising in terms of being accepted by a regulatory authority?
Oh, okay. That's a good question. Thank you very much. Okay, so I don't see any other method for the total bacterial count, but
Mm,
for specific pathogens, the many regulatory agency adopt PCR method, and sometimes they also adopt ELISA method.
mm,
Because specific pathogen, they only check the presence of absence.
mm.
So
I see.
PCR or ELISA method can easily show that the result of like a presence of absence of pathogenic bacteria.
I see. Okay. And are they currently accepted by the regulatory authorities, Dr. Yu, to detect bacterial count, or it's
Sorry, can
something
you?
that is uh, have they been accepted, the ELISA and the PCR methods? Are they acceptable techniques that we can use?
That that's I just give a general example of the
Okay.
question. Yeah, so
Yeah.
because each regulatory agents they just see that any method equivalent to the like a culture-based method, they are
Mm.
adopted method. So the
Okay.
either well, PCR or ELISA, they show the same results as the culture-based method. They are adopted.
Hmm. I see. Okay. Thank you for the answer. And just for the last question, how do you differentiate between the presence of probiotics and undesirable bacteria or pathogenic bacteria in foods, since the bacterial count methods of measuring the quantity or the presence of bacteria seem to be quite general?
You mean how to differentiate between the probiotics
Yes, that the
and
healthy
the pathogen?
bacteria versus those that are pathogenic. Yeah.
Oh, okay, that's a good
Yeah.
question.
Okay.
Well, it's probiotics, normally lactase bacteria.
Mm.
Their characteristic is totally different with the pathogenic bacteria.
Yeah.
So the most pathogenic bacteria, like for example Salmonella, we can based on culture-based method, we can use the different types, selective agar, selective broth, which are
Mm.
using their some using their fermented product.
Mm.
So in that case, we can easily differentiate them. Or if you use the PCR method, we can use that specific primers, which
Mm.
only detect the
Yeah.
specific gene and a certain pathogen.
Mm. I
So
see.
the
Okay.
lactase bacteria and the specific pathogen, they are totally different metabolites. We can use this metabolite to differentiate them.
Yeah. Okay. Yeah, that makes sense. Thank you so much, Dr. Yu. And I think that is all the questions that we have. If you do have any other questions, you're more than welcome to send us an email, and we can collate all these questions and then discuss again with Dr. Yu in 2 weeks' time. So next week there will be another lecture, but Dr. Yu will not be joining us live. Okay, so with that, I would like to thank Professor Yu for joining us live today. Thank you, Prof. Yu, and we will see you in 2 weeks' time again for the last lecture.
Okay, thank you very much. See
Okay,
you
thank
and
you,
next
Prof. Yu.
time.
Okay, see
Bye.
you. Bye bye. Thank you so much. Yeah. And for the rest of the participants who would like to get your help to hopefully complete our survey by scanning the QR code or going through the URL link that has been pasted on the chat box. And at the end of the survey, there will be a link that you can click to get our instant certificate of participation. And if you do have any issues to get the certificate, please do let us know by sending us an email, and then we will reply to you as soon as possible, and we'll do our best to make sure that the certificate is issued to you if you have actually completed our survey. And with that, we would like to thank you for your participation, and we look forward to having you again next week for another exciting lecture from Professor Yu. Have a good afternoon and good lunch. Thank you.